Hello everyone. Welcome to the second part of our series on genetic material based diagnosis for viruses. As we continue our exploration into diagnostic methodologies, today's focus centers around polymerase chain reaction (PCR) techniques, which stand at the forefront of molecular diagnostics for viral infections. PCR has become an indispensable tool in the realm of viral diagnosis due to its remarkable sensitivity, specificity, and versatility. In this session, we delve deeper into various PCR techniques tailored for detecting viral genetic material, from traditional methods like endpoint PCR to advanced techniques such as digital droplet PCR and quantitative PCR. We'll unravel their principles, applications, and the pivotal role they play in diagnosing viral infections accurately and efficiently. Unveiling the power of polymerase chain reaction (PCR). The PCR technique was developed in the late 1980s by Kerry Mullis and has been extensively applied in diagnostic virology. PCR is a highly sensitive technique and requires minimal template for detection and amplification of specific sequences. Applications of PCR techniques in viral diagnostics. PCR techniques play a crucial role in viral diagnosis across various applications: detection of viral pathogens in clinical samples, monitoring viral load and disease progression, genotyping and subtyping of viral strains, surveillance of emerging viral diseases, evaluation of antiviral drug efficacy and resistance. Now let us look into the key components required to set up a polymerase chain reaction. For standard PCR, all you need is a DNA polymerase. DNA polymerase is the enzyme responsible for synthesizing new DNA strands by adding nucleotides to the growing chain. Commonly used DNA polymerases include Taq polymerase, PFU polymerase, and clenofragment. Magnesium ions. Magnesium ions are cofactors required by DNA polymerase for its enzymatic activity. They stabilize the DNA polymerase DNA complex and facilitate nucleotide addition. DNTPs or nucleotides. Nucleotides, also known as deoxynucleoside triphosphates, are the building blocks of DNA. They consist of adenine, thymine, cytosine, and guanine. Primers. Primers are short, single-stranded DNA sequences that anneal to the target DNA sequence. They provide the starting point for DNA synthesis by DNA polymerase. Primers are designed to flank the region of interest and define the boundaries of the amplified fragment. DNA template. The DNA template is the target sequence that we want to amplify. It can be genomic DNA, plasmid DNA, cDNA, or any other DNA source containing the target sequence. Thermocycler. A thermocycler is a specialized instrument used to automate the temperature cycling process in PCR. It provides precise control over temperature changes required for denaturation, annealing, and extension steps of PCR. Now let us quickly go through the steps of a polymerase chain reaction. Thermal cycling usually comprises of three steps, which are denaturation, annealing, and extension. Denaturation, whereby the double-stranded DNA template is heated to separate it into two single strands. The denaturation temperature ranges between 94 to 96 degrees centigrade depending on the type of the template. Second step is the annealing, where the temperature is lower to enable the DNA primers to attach to the template DNA. Annealing usually ranges from 55 to 60 degrees centigrade depending upon the set of the primer and the template DNA. Extension. Extension is where the temperature is raised to allow the copying of the new strand of DNA by the Taq polymerase enzyme. Elongation usually occurs at 70 to 72 degrees centigrade. These steps occur sequentially over a series of temperature and timings. Each step of the PCR cycle must be optimized for each template and the primer set used. The PCR mixture undergoes amplification in a thermal cycler until all the desired cycles are completed. The cycle is repeated approximately 20 to 40 times. A portion of the tube's content is typically analyzed using gel electrophoresis and it's stained with Ethidium bromide dye. Agarose gel electrophoresis separates the DNA fragments in a mixture according to their sizes. The smaller fragments move farther through agarose gel than the larger fragments. Ethidium bromide binds to DNA and fluoresces when excited under a UV light. If the amplification was successful, then a product called the amplicon can be detected as a band on the agarose gel. Now let us look into some variations of the conventional PCR. Nested PCR. In nested PCR, two rather than just a single pair of primer target a single locus. The first pair amplifies a target fragment in a conventional PCR reaction. The second pair anneals to the sites within the first amplicon and amplifies an internal shorter segment. The purpose of nested PCR is to increase the assay sensitivity by re-amplifying the target from a template previously enriched by the first PCR. Non-target sequences amplified non-specifically in the first PCR are not re-amplified in the second reaction as they would be unlikely to possess the internal priming sites targeted by the second PCR. Nested PCR procedures suffer from longer turnaround times. They are difficult to automate. Now let us look into one more variation of the conventional PCR, the multiplex PCR. In multiplexing assay, more than one target sequence can be amplified using multiple primer pairs in a reaction mixture. As an extension to the practical use of PCR, this technique has the potential to produce considerable savings in time and effort within the laboratory without compromising on the utility of the experiment. Types of multiplex PCR. Single template PCR reaction. This technique uses a single template which can be a genomic DNA along with several pairs of forward and reverse primers to amplify specific regions within a template. Multiple template multiplex PCR reaction. It uses multiple templates and several primer sets in the same reaction tube. Presence of multiple primers may lead to cross-hybridization with each other and the possibility of mispriming with other templates. Advantages of multiplex PCR. Potential problems in a simple PCR include false negatives due to the reaction failure or false positives due to the contamination. False negatives are often revealed in multiplex assays because each amplicon provides an internal control for the other amplified fragments. Efficiency. The expense of reagents and preparation time is less in multiplex PCR than in systems where several tubes of uniplex PCRs are used. A multiplex PCR reaction is ideal for conserving costly polymerase and templates in short supply. Indication of template quality. The quality of template may be determined more effectively in multiplex than in a simple PCR reaction. Indication of template quantity. The exponential amplification and internal standards of multiplex PCR can be used to assess the amount of a particular template in a sample. To quantitate templates accurately by multiplex PCR, the amount of reference template, the number of reaction cycles, and the minimum inhibition of the theoretical doubling of product for each cycle must be accounted. Some viruses have RNA as their genetic material. How can such viruses be diagnosed using the polymerase chain reaction? Reverse transcriptase PCR is a modification of the standard PCR technique that can be used to amplify mRNA. The first step is to convert the isolated mRNA into a complementary DNA known as the cDNA molecule, which is prepared using RNA-dependent RNA polymerase enzyme, also known as the reverse transcriptase enzyme, during a process called reverse transcription. The complementary DNA can be used as any other DNA molecule for PCR amplification. The primers used for cDNA synthesis can either be a non-sequence specific primer, that is, a mixture of random hexamers or oligo-DT primers or sequence specific primers. Random hexamers are mixtures of all possible combinations of 6 nucleotide sequences that can attach randomly to mRNA and initiate reverse transcription of the entire RNA pool. Oligo-DT primers are complementary to the poly-A tail of mRNA molecules and allow synthesis of cDNA only from mRNA molecules. Amplification of a housekeeping gene must accompany each reverse transcriptase PCR reaction as an internal control to monitor the quality and quantity of RNA in a given sample. Endpoint PCR is a method for nucleic acid amplification where the maximum number of copies of DNA or RNA target is produced. This type of PCR offers qualitative answers on whether or not a particular target sequence is present, but not quantitative information. RT-PCR is also a form of endpoint PCR like the conventional PCR. Real-time PCR. In conventional PCR, the amplified DNA product or amplicon is detected in an endpoint analysis. In real-time PCR, the accumulation of amplification product is measured as the reaction progresses in real time with the product quantification after each cycle. It is a powerful technique that offers great sensitivity and specificity and can be used for both quantitative and qualitative analysis. Real-time PCR is also known as quantitative polymerase chain reaction. Reverse transcriptase quantitative PCR is used for diagnosis of viruses having RNA as their genetic material. Two methods are available for quantification of gene expression by real-time qPCR, two-step RT-qPCR and one-step RT-qPCR. In both cases, RNA is reverse transcribed into cDNA and the cDNA is then used as a template for qPCR amplification. One-step and two-step refer to whether the reverse transcription and real-time PCR amplification are performed in same or separate tubes. In the two-step method, RNA is first transcribed into cDNA in a reaction using reverse transcriptase. An aliquot of the resulting cDNA is then used as a template for multiple qPCR reactions. In the one-step method, reverse transcription and qPCR are performed in the same tube. Detection methods in qPCR. Two methods are commonly used for detection and quantification in qPCR. One is the cybergreen method, other is the Tachman probe-based method. cybergreen method is based on a non-specific dye, usually cybergreen, that causes fluorescence by binding to amplicons and other double-stranded DNA fragments in the qPCR reaction. The increase of amplicons is proportional to an increase in fluorescence intensity, which is measured at each PCR cycle. The result of the experiment depends on the specificity of the primers used in the PCR reaction. In case of non-specific binding or amplification, the dye will also bind to non-specific double-stranded DNA and emit fluorescence. It can even give us a positive signal in the case of primer dimers as well. Thus, this technique is less sensitive compared to the probe-based technique. Tachman probe-based method. Tachman PCR uses a dye-labeled nucleic acid probe complementary to an internal segment of the target DNA. This dye-labeled probe anneals to one of the template close to and downstream from one of the two PCR primers. The probe is labeled by two fluorescent moieties. The reporter fluorophore is attached to the probe's 5' end and the quencher is attached to the 3' end. When the reporter and quencher are connected, the quencher reduces the fluorescence of the reporter dye as it absorbs energy via a fluorescence resonance energy transfer FRET. However, during PCR, the polymerase extending the primer on the probe's target strand displaces and degrades the annealed probe through the action of 5' to 3' exonuclease function. The fluorophore is released from its molecular attachment to the quencher and starts fluorescing. As more PCR products are generated, the more dye-labeled probe will find target regions to join, which eventually leads to the release of reporter molecule during subsequent amplification process. The release of reporter dyes is reflected as increased intensity of the fluorescence signal, which is proportional to the amount of amplicon synthesized. Now, let us quickly go through some basics of real-time PCR data analysis. In this plot, the number of PCR cycles is shown on the x-axis and the fluorescence from the amplification, which is proportional to the amount of amplified product in the tube, is shown on the y-axis. The plot represents a sigmoidal amplification pattern. This sigmoidal curve demonstrates the change in fluorescence over the number of cycles. Typically, in a qPCR experiment, there are 40 cycles. That is, there are 40 rounds of amplification. At the start of the run, when the amount of PCR product is low, that is during the initiation phase, it produces very less fluorescence. This section of cycles between cycle 0 to 15 is known as the background signal or the baseline signal. Next, the exponential phase. Once the amount of PCR product is amplified enough, it will enter the exponential phase. This is when the amount of PCR product doubles every PCR cycle. This can be seen in cycles 15 to 25 in this figure. Plateau phase. Once all of the reagents such as the nucleotides have been used up in the PCR reaction, the amplification will slow and ultimately plateau. This is the region where no more PCR products are produced. This can be seen in cycles 30 to 40 in the above amplification curve. What is CT value? The terms cycle quantification CQ, crossing point CP or takeoff point TOP refer to the same value as the CT value, the threshold cycle value. The CP, crossing point PCR cycle, or CT, cycle threshold value, is the cycle at which the fluorescence achieves a defined threshold. It corresponds to the cycle at which a statistically significant increase in fluorescence is first detected. This concept is the basis for accurate and reproducible quantification using qPCR. The number of cycles needed for the amplification associated fluorescence to reach a specific threshold level of detection, the CT or CP value, is inversely correlated to the amount of nucleic acid that was in the original sample. The value always is in the exponential phase of amplification, when amplification is most efficient and therefore quantification is least affected by reaction limiting conditions. In order to calculate the CT value, it is necessary to draw a horizontal line threshold on the amplification plot. The placement of the line is often determined by the qPCR software. However, users can manually place this line. Ideally, the threshold is placed at a point when the reaction is in the exponential phase. Therefore, it will not be confused with the background signal. In this example, it can be seen that the cycle number when the threshold value intersects the amplification plot is 19. Therefore, the CT value will be 19 in this case. The CT value is associated with the amount of PCR product in the reaction. The lower the CT value, the more the product that is present. This is because it takes fewer PCR cycles for that product to be detected over the background signal. Dissociation curve of real-time PCR. The main advantage of quantitative PCR is the amplicon validation through melting curve analysis. We can quantify how many amplicons are generated and how many non-specific primer-dimers are formed during PCR reaction by doing the melting curve analysis. Melting curve analysis is a type of endpoint detection that quantifies the templates after the completion of the reaction. This type of analysis is commonly used for dye-based real-time PCR for accurate quantification. So after the completion of the reaction, the temperature is gradually increased to melt the double-stranded DNA, meaning the copies of the double-stranded DNA generated are now melted or separated. Eventually, the dye dissociates from the double-stranded DNA and the fluorescence signals gradually decrease. A single and sharp peak for all the templates in the graph indicates specific amplification, while rest of the peaks indicate non-specific products or formation of primer dimers. The real-time PCR technique has emerged as a gold standard for COVID-19 diagnosis. A cotton swab is inserted into the patient's nose or throat to collect the respiratory secretions. The sample may contain virus particles if the individual is infected with SARS-CoV-2, the virus responsible for COVID-19. The collected specimen can be stored at 2 to 8 degrees for up to 72 hours. The collected sample undergoes a process to deactivate the virus while simultaneously extracting RNA from it. This step is crucial for ensuring the safety of the laboratory personnel handling this sample and for facilitating subsequent RNA analysis. Reverse transcriptase PCR. The purified RNA strands are then subject to reverse transcription, wherein the viral RNA is converted to complementary cDNA using an enzyme, reverse transcriptase. This step is necessary because PCR amplification works with DNA and not RNA. The cDNA is then subject to PCR amplification. In real-time PCR, the amplification process is monitored in real time using fluorescent dyes or probes. This allows for detection and quantification of target gene sequences. The target gene sequences that are used in COVID-19 RT-PCR include the RDRP gene, that is, the RNA-dependent RNA polymerase gene. This gene encodes the RNA polymerase enzyme, which is essential for replicating the viral genome. It is a highly conserved region and is commonly targeted in COVID-19 RT-PCR assays. The ORF1A gene is a part of the viral replicase complex and is also targeted in many COVID-19 RT-PCR assays due to its conserved nature. The E gene, or the envelope protein gene, this gene encodes the envelope protein of the virus and is another common target in COVID-19 RT-PCR assays. It is often targeted alongside other genes for increased sensitivity and specificity. The test results are obtained shortly. CT value. In real-time PCR analysis, the cycle threshold value corresponds to the cycle number at which the fluorescence signal of the amplification reaction crosses a predetermined threshold. Lower CT values indicate a higher concentration of the target gene in the sample, suggesting a higher viral load. The CT value can be used to semi-quantitatively estimate the amount of viral RNA present in the sample and to compare the viral loads between different samples and individuals. Droplet digital PCR is an innovative and promising approach for highly sensitive quantification of nucleic acids. The principle of this method is based on water-oil emulsion droplet technology, in which the sample is partitioned into 20,000 droplets, each representing a microreactor for individual PCR reaction in which the presence of target is revealed by the emission of fluorescence. This results in the separation of positive droplets containing at least one copy of the target and exhibiting increased fluorescence, and negative droplets, both individually counted on a dedicated droplet fluorescence reader. Lastly, the fraction of positive droplets is fitted to a Poisson distribution to determine the absolute starting copy number in the sample. Each reaction in a dPCR can be thought of as a binary event. Either it contains the target molecule positive or it does not, negative. This digital aspect of ddPCR is akin to digital electronics, where information is represented as discrete states, 0 or 1. Therefore, ddPCR provides a digital readout of the presence or absence of the target molecule in each reaction. The process of ddPCR involves multiple steps. First, the sample is prepared by extracting the nucleic acid and purifying it. Prior to droplet generation, ddPCR reactions are set up in the same way as the real-time qPCR reactions that use a Tachman hydrolysis probe labeled with reporter fluorophores or any intercollecting dye such as cybergreen or eva green in a 96 well plate. The plate containing the reactions is then placed in an automated droplet generator that divides each reaction into thousands of small droplets. Approximately 20,000 nanoliter sized droplets. This step generates uniform droplets containing a small amount of template DNA or RNA, enabling precise target quantification. The droplets are then transferred to a 96 well plate and placed in a thermal cycler for amplification. Post amplification, the plate is placed in a droplet reader to analyze individual droplets using either 2-color or 6-color detection. system. Positive droplets containing at least one copy of the target DNA molecule exhibit increased fluorescence compared to negative droplets. The data can be viewed using proprietary software in a 1D or 2D plot. Now let us quickly explore the differences between endpoint PCR, qPCR and ddPCR. Differences in methodology. In endpoint PCR, the amount of product is measured after amplification of target nucleic acid molecules in one reaction chamber. In qPCR, the amount of product is measured during amplification of all target nucleic acid molecules in one reaction chamber. In ddPCR, the amount of product is measured after amplification of individual copies of target nucleic acid molecules in its own reaction chamber. Advantages of endpoint PCR is that it provides simple yes-no qualitative answers. Advantages of qPCR is that it is active over a broad dynamic range of approximately 7 logs. It is quantitative in nature. Detection is capable down to a two-fold change, and there is no post-PCR processing. In case of ddPCR, it is an absolute quantification. There is no need for reference standards or endogenous controls. It is less sensitive to inhibitors. Detection can measure a 30% smaller difference than qPCR. The disadvantages of endpoint PCR is that there is poor precision, low sensitivity, narrow dynamic range of less than 2 logs, not quantitative, and it requires post-PCR processing. The disadvantages of qPCR are that it requires reference standards or endogenous controls, and it is sensitive to inhibitors. The disadvantages of ddPCR is that it is more expensive than qPCR and it works over a narrow dynamic range than qPCR. ddPCR has successfully been employed for detecting SARS-CoV-2 virus in wastewaters. Wastewater-based epidemiology indirectly monitors infection rates in a community based on pathogen levels in local wastewater. Because symptomatic and asymptomatic SARS-CoV-2 infected individuals shed viral RNA in sewer systems, analysis of SARS-CoV-2 in sewage networks can provide information about SARS-CoV-2 infections within the community. However, the accuracy of these data depends on the quality of the wastewater testing method. During the COVID-19 pandemic, droplet digital PCR has proven that it has the sensitivity needed to detect outbreaks and monitor SARS-CoV-2 variants in communities via wastewater testing. Recently, the first verified cure of a child with HIV was reported. After conventional tests detected no virus, the ddPCR system provided a more sensitive assay to confirm that the child was functionally cured and there was no latent viral reservoir. Now let us discuss some MCQs on these PCR techniques. Which PCR method is suitable for detecting low copy number DNA sequences by amplifying a target region within 2 rounds of PCR? A. Endpoint PCR, B. Digital droplet PCR, C. Quantitative PCR, or D. Nested PCR. The correct answer is D. Nested PCR. Which PCR technique utilizes a special molecule that generates fluorescence during DNA amplification, allowing real-time monitoring of the reaction? A. Endpoint PCR, B. Digital droplet PCR, C. Quantitative PCR, or D. Nested PCR. Answer is C. Quantitative PCR. Which PCR variant involves the use of multiple primer sets in a single reaction to amplify multiple targets simultaneously? A. Endpoint PCR, B. Digital droplet PCR, C. Quantitative PCR, or D. Multiplex PCR. Answer is D. Multiplex PCR. Which PCR method is employed when high sensitivity and specificity are required, especially for detecting rare targets? A. Endpoint PCR, B. Digital droplet PCR, C. Quantitative PCR, or D. Nested PCR? Answer is B. Digital droplet PCR. In which PCR method is the endpoint of a reaction determined by gel electrophoresis to visualize the amplified DNA fragments? A. Endpoint PCR, B. Digital droplet PCR, C. Quantitative PCR, or D. Nested PCR? Answer is A. Endpoint PCR. These are a few of the references. Thank you for watching.